Hello everyone, and welcome to our channel, Mootechie. A warm welcome to all the dairy technologists and students tuning in. Today we are embarking on an exciting journey through the world of industrial microbiology and its many products. Let's begin with section 15.1, Industrial Products and the Microorganisms that Make Them. Industrial microbiology is the field that uses microorganisms, typically grown on a very large scale, to produce valuable products or carry out specific chemical transformations. This field originated with ancient processes like alcoholic fermentation. Later, more complex processes were developed for producing pharmaceuticals, food additives, enzymes, and various chemicals. The major organisms used in these processes are fungi and bacteria from the genus Streptomyces. To improve production, classic methods are used to select for high-yielding microbial variants. Now, what makes a microbe useful for industry? The ideal industrial microbe has several key properties. It should produce spores or be easily inoculated into a fermenter. It must grow rapidly on a large scale in an inexpensive growth medium. It should produce the desired product quickly. For safety, it should not be pathogenic. And finally, it should be amenable to genetic manipulation to further improve its capabilities. Moving on, we will see the diverse range of microbial products of industrial interest. These include the microbial cells themselves, which can be used as a product. They also include enzymes, antibiotics, steroids, alkaloids, and various food additives. A major category is commodity chemicals. These are inexpensive chemicals produced in very large quantities. Examples include ethanol and citric acid. Next, we explore section 15.2, Production and Scale. It's important to distinguish between two types of microbial products. A primary metabolite is produced during the exponential growth phase of the microorganisms. A classic example is alcohol. In contrast, a secondary metabolite is produced during the stationary phase, after the main growth period has slowed down. Let's delve deeper into secondary metabolites. These compounds are not essential for the basic growth of the microbe. Their formation depends heavily on the specific growth conditions. They are often produced as a group of related compounds, rather than a single substance. A key feature is that they are often significantly overproduced by the organism. They are frequently produced by spore-forming microbes during the process of sporulation. A figure 15.1 helps illustrate this. Part A shows a graph for a primary metabolite like alcohol. As the sugar is consumed and the number of cells increases during exponential growth, the alcohol is produced. Part B shows a graph for a secondary metabolite like penicillin. Here, the sugar is consumed and cell growth occurs first. The production of penicillin only begins later, during the stationary phase, after the main growth has finished. Another important point about secondary metabolites is that they are often large, complex organic molecules. Their production requires a large number of specific enzymatic steps. For example, the synthesis of the antibiotic tetracycline requires at least 72 separate enzymatic steps. The starting materials for these complex molecules arise from the major biosynthetic pathways within the cell. Now let's talk about where this happens: the fermenter. The fermenter is the vessel where the microbiological process takes place, as shown in figures 15.2a and b. In industrial microbiology, any large-scale reaction carried out by microbes is referred to as a fermentation, and most of these are aerobic processes requiring oxygen. Fermenters vary enormously in size, from small 5-liter lab units to enormous vessels holding 500,000 liters. There are designs for both aerobic and anaerobic processes. Large-scale fermenters are almost always constructed from stainless steel. Inside, impellers and spargers work together to supply and mix oxygen into the culture broth, which is depicted in figure 15.2c. Figure 15.2a shows a photograph of a large, industrial-scale fermenter. Figure 15.2b provides a detailed schematic diagram of a fermenter. It labels key components like the steam inlet, a viewing port, the impeller for mixing, a cooling jacket, ports for external cooling water in and out, a motor, a sterile seal, a harvest pipe, a pH controller, acid-base reservoirs and pumps, an air filter, an exhaust, the culture broth, and a sparger that introduces sterile high-pressure air for aeration. Figure 15.2c shows a close-up photograph of an impeller, the mixing device inside the fermenter. Another figure, 15.3, shows an image of a pilot-scale fermenter used for research and development. Coming up, we discuss section 15.3, Antibiotics, their isolation, yield, and purification. Antibiotics are compounds that kill or inhibit the growth of other microbes. They are typically secondary metabolites. Most antibiotics used in clinical medicine are produced by filamentous fungi or by actinomycetes bacteria. New antibiotics are still discovered through laboratory screening, as shown in Figure 15.4a. In this process, microbes are obtained from natural environments like soil and isolated into pure culture. They are then assayed or tested to see if they produce substances that inhibit the growth of test bacteria. Figure 15.4a illustrates the first step in antibiotic discovery, which is isolation. A dilution of a soil sample is spread onto a plate of selective medium using a sterile glass spreader. After incubation, colonies of microbes like Streptomyces grow. To find which ones produce antibiotics, the plate is overlaid with a layer of an indicator organism, like a test bacterium. After another incubation, zones of growth inhibition appear around the colonies that are producing organisms, indicating where the antibiotic has diffused into the agar and killed or inhibited the test bacteria. A common method for testing new isolates is the cross-streak method, shown in Figure 15.4b. The antibiotic-producing microbe is streaked across one side of a plate and allowed to grow and produce its antibiotic, which diffuses into the agar. Then different test organisms are streaked perpendicularly to the original streak. After incubation, the growth of the test organisms is observed. Inhibition zones appear where the test organisms are sensitive and could not grow near the antibiotic, diffusing from the producer streak. Unfortunately, most new isolates produce antibiotics that are already known. Furthermore, most new antibiotics fail subsequent toxicity and therapeutic tests in animals. The time and cost of developing a new antibiotic to market are enormous, approximately 15 years and $1 billion. A process that involves rigorous clinical trials and approval from bodies like the US Food and Drug Administration. The purification and extraction of an antibiotic often involve very elaborate and precise methods. In the following section, 15.4, we look at the industrial production of two famous antibiotics, penicillins and tetracyclines. Penicillins are a class of beta-lactam antibiotics. There are natural penicillins, biosynthetic penicillins, and semi-synthetic penicillins, as shown in Figure 15.5. The production of penicillin is a classic example of a secondary metabolite. Production only begins after the carbon source in the medium is nearly exhausted, which is illustrated in Figure 15.6. Interestingly, high levels of glucose actually repress penicillin production. Figure 15.6 is a graph showing the progress of a penicillin fermentation over time. It tracks biomass, carbohydrates like lactose, ammonia levels, and penicillin production. Initially, cell biomass increases as lactose is consumed. Penicillin production begins much later, after the main growth phase, and is sustained through feeding with glucose and nitrogen. The biosynthesis of tetracycline is incredibly complex, involving a large number of enzymatic steps. There are more than 72 known intermediates in the pathway. Astonishingly, more than 300 genes are involved in this process. This indicates a very complex system of biosynthetic regulation. As we continue, we find section 15.5 on vitamins and amino acids. The production of vitamins by microorganisms is a huge industry, second only to antibiotics in terms of total pharmaceutical production. Vitamin B12, for instance, is produced exclusively by microorganisms. A deficiency in B12 results in pernicious anemia. This vitamin is unique because it contains the element cobalt. Another vitamin, riboflavin or B2, can also be efficiently produced by microbes. Amino acids are another major microbial product. They are used as feed additives in the food industry, as nutritional supplements in the nutraceutical industry, and as starting materials in the chemical industry. Important examples include glutamic acid, which is used to make monosodium glutamate, or MSG. Aspartic acid and phenylalanine are used to produce the sweetener aspartame, known as NutraSweet. Lysine is another important amino acid produced on a large scale for use as a food additive. Now let's look at section 15.6, enzymes as industrial products. Many industrially useful enzymes are exoenzymes. These are enzymes that the microbe excretes into the surrounding medium instead of keeping them inside the cell. They are extracellular. This is useful because they can digest large, insoluble polymers, like cellulose, protein, and starch. Enzymes are fantastic industrial catalysts, because they typically produce only one specific stereoisomer of a product and have very high substrate specificity, meaning they act on only one particular compound. Figure 15.10 shows two images related to industrial enzymes. Part A is a photo of a bag containing an enzyme system designed for corn and soy-based poultry feed, produced by a company called FinFeeds. The bag has a net weight of 25 kilograms. Part B is a graph showing the stability of an enzyme called pullulanase over time. The y-axis shows the % of enzyme activity remaining, and the x-axis is time in hours. The graph demonstrates how the enzyme's activity decreases over time, which is important for determining its shelf life and effectiveness. Moving on to part 3 of our presentation, we explore alcoholic beverages and biofuels. This includes the topics of wine, brewing and distilling, and biofuels. First, section 15.10 covers wine. Most wine is made from the fermentation of grapes. Wine fermentation takes place in fermentors that can range in size from 200 to 200,000 liters. These fermentors can be made from various materials, including oak, cement, glass-lined steel, or stone, as shown in figures 15.12b, c, and d. White wine is made from the juice of white grapes, or from red grapes that have had their skins removed immediately, which is illustrated in figure 15.13. Red wine is typically aged for months or even years to develop its flavor, while white wine is often sold without much aging. Figure 15.12b shows an image of large oak barrels used for wine fermentation and aging. Figure 15.12c is a photograph of the Christian Brothers Winery, showing the scale of their operation. Next, in section 15.8, we discuss brewing and distilling. Brewing is the term used to describe the manufacture of alcoholic beverages, like beer, from malted grains. This process is shown in figure 15.14. Yeast is the microorganism used to produce beer. There are two main types of brewery yeast strains. Top fermenting yeasts are used for ales, and bottom fermenting yeasts are used for lagers. Figure 15.14 includes several images provided by Bush Creative Services, part of the Anheuser-Busch Company, showing different aspects of the brewing process. Distilling is a process used to make spirits. Distilled alcoholic beverages are produced by heating a previously fermented liquid. The heat volatilizes or turns into vapor most of the alcohol. This alcohol vapor is then collected and condensed back into a liquid, resulting in a much higher alcohol content. Examples include whiskey, rum, brandy, vodka, and gin. This process is illustrated in figure 15.16. On an industrial scale, over 50 billion liters of ethanol are produced yearly for non-beverage purposes. It is used as an industrial solvent and as a supplement for gasoline. Following this, we examine section 15.9 on biofuels. Ethanol is a major biofuel and industrial commodity chemical. Over 60 billion liters are produced yearly from the fermentation of agricultural feedstocks like corn and sugarcane, as shown in figures 15.17a and b. This ethanol is blended with gasoline to create gasohol or used in higher concentrations in fuels like E85. The field also includes petroleum biofuels, which involves the microbial production of alternatives like butanol. Another exciting area is the synthesis of petroleum-like compounds directly from green algae, which is depicted in figure 15.17c. Figure 15.17 shows images related to biofuel production, including a field of feedstock and an algae biofuel reactor. Finally, we reach part 4, products from genetically engineered microorganisms. This covers topics like expressing mammalian genes in bacteria, producing engineered somatotropin, other mammalian proteins, engineered vaccines, genome mining, and engineering metabolic pathways. Section 15.10 introduces the concept of biotechnology, which is the use of living organisms for industrial or commercial applications. A key tool is the genetically modified organism, or GMO, which is an organism whose genome has been deliberately altered. Genetic engineering allows us to express eukaryotic genes, like human genes, in prokaryotes like bacteria. A famous example is the production of human insulin in E. coli. This is achieved by cloning the gene, often starting with its messenger RNA, as shown in figure 15.18, or by finding the gene based on knowledge of the protein it produces. Thank you for joining this comprehensive overview of industrial microbiology. We've covered everything from the microbes themselves to the vast array of products they create, including antibiotics, vitamins, enzymes, alcoholic beverages, biofuels, and the revolutionary field of genetic engineering. The ability to harness these tiny organisms has a profound impact on our world. Thank you. See you in the next class.